This is David Henderson from the Clan Henderson Society DNA project. In this video, we'll discuss how single nucleotide polymorphisms and the haplotree are useful in genetic genealogy. First of all, what are single nucleotide polymorphisms, which we'll call SNPs? They're basically spelling errors. The DNA chain is made up of 4 letters, A, C, G, and T. If in the process of making a duplicate of the DNA, an A gets converted to a C, and that change persists, that becomes a SNP, and it can persist for forever, basically. There are billions of DNA letters in the human DNA genome, and there are at least 1 million SNPs that have been observed. So each of us carries a lot of these spelling errors with us. The process of copying DNA every time a cell divides is very prone to errors. There are a lot of spell checkers built into us so that the actual number of errors is less than would be expected from the copying. But the fact is that a lot of these errors are generated, and each new generation can generate 1 or more new errors. Even 2 brothers can have different SNPs because their father is aging between their births. The Clan Stuart Society has discovered that the DNA of the Royal Stuart line from one of the Stuart kings has a difference in SNPs between 2 sons. So between the birth of the first son and the birth of the second son, a mutation occurred in the father, which produced a different SNP in the second son. While there are about 1 million of these SNPs in the human DNA genome, only about 20,000 of these have been characterized so far. It takes a lot of work to characterize these SNPs, and I don't fully understand how this is done. Some of it is done by studying DNA of fossils and DNA of bodies that were buried thousands or tens of thousands of years ago. Some of it is done by studying where people live now and what SNPs they carry. But the process of developing the human haplotree involves trying to determine where these SNPs occurred and when they occurred. And this is an ongoing process, so researchers who study this are constantly adding SNPs and extending the haplotree. So on the right we see an image of the human haplotree. The haplotree starts in Africa, where the first modern humans evolved. And this is called Group A. At some point along the way, a mutation occurred, which is called M42, and that mutation divided the human race into 2 groups. The A group that did not have that mutation divided the human race into 2 groups. One that did not have the M42 mutation, which remained Group A, and one group that had the mutation, which became Group B. Later, a mutation at the position M168 separated Group B from Group C. And this process continued, and the mutations are identified by a letter and number system. So here we see groups A through S. So within the Henderson project, we've identified 5 haplogroups that are present. One is not shown here. We have some Hendersons that have the E haplogroup, and these are Hendersons of African American lineage. We have just a small number of G haplogroup individuals, a small number of J haplogroup individuals. About 20% or a little bit less of the Hendersons that have been tested so far fall in the I haplogroup. This group carries the M107 mutation, and the rest of the Henderson group, which is about 80% of everyone we've tested so far, fall in the R haplogroup. The R haplogroup is defined by the M207 mutation, but only one family group out of the 20 that we've found so far do not carry the M269 mutation. So the vast majority of all Hendersons have the M269 mutation. So when you have your DNA tested at Family Tree DNA for Y-DNA testing, they will assign a haplogroup based on the STRs in the DNA. The STR groups are associated with haplogroups, and it is possible to get at least a crude idea of the haplogroup of a person based on their STR results. And so this allows Family Tree DNA to separate people who presumably have the I-170 mutation or the R269 mutation. The I-170 mutation is primarily thought to be Viking and Scandinavian, although there are also some with this that lived in southeastern Europe. This mutation arose during the last glacial maximum and is associated with a group of hunter-gatherers who lived in Paleolithic Europe. The M269 mutation is much newer, having occurred somewhere between 4,000 and 9,000 years ago. It's associated with the Neolithic expansion from Central Europe and Asia. So the I haplogroup is associated with a group of hunter-gatherers who lived in Europe in the glacial maximum, whereas the R haplogroup is associated with early farmers who moved from Central Europe and Asia into Europe about 4,000 years ago. These are both very old mutations and not useful at all for genealogy. In order to get more useful information, it's necessary to do deeper testing to reveal a terminal haplogroup.